Dr. Mark Hannigan from Virginia Tech University, the floor is all yours.
Thank you. I was looking through the list of people that are attending or semi-attending, I guess, and it's quite an eclectic mix from all over the world. So it's interesting to see all the names there. But today, my talk probably seems to deviate a little bit from the overall conference topic, but I think it's an important one that Heather's brought out in terms of methionine and some of these other precursors being important relative to one carbon metabolism. So, of course, to understand what's available for one carbon metabolism, we need to understand, what the supply is of some of those precursors. So I'm going to focus today on, sort of assessing the amino acid supply in ruminant species. And I want to acknowledge it up front, co-authors of Carrie Estes, Xinbei Wang, Jacqueline Prestegard. They've conducted a bunch of the work that I'm going to present today, and so a lot of these ideas, originate from their work. So, of course, there's lots of reasons, other than just one carbon metabolism, or at least most reasons that we're interested in nutrition, eventually come back to money. And so when you look at the relative cost of feeding a dairy cow based on 5-year averages for nutrient prices as assessed by the Ohio State Group, you find that on average, the metabolizable protein supply will cost about 40% of the cost of the diet. So in this case, for 5-year average to feed an 80-pound cow, it's going to cost about $2.34 a day. Now, if you take that 80-pound cow at 3% protein, that's about 2.4 pounds of protein. So you can take 2.4 divided by 5.4 and see that we're only capturing about 35 to 40% of the metabolizable protein that we're putting in. And of course, we're not capturing all of the crude protein and metabolizable protein. So the amount of protein that's being sort of wasted or being inefficiently used is significant in dairy cattle, and it's actually similar in beef cattle as well. So the question, that has driven our research for a number of years is whether or not this is, a requirement to be a ruminant, that no ruminants can actually do better than this, or is this simply an artifact of the way that we're feeding? And so I think the answer, won't be in today's talk, but essentially, I think all of us feel like it's the way we're feeding. And so the way to sort of approach that or to solve that problem is to try to gain a better understanding of what the true requirements for the amino acids are, because the cow doesn't have a requirement for protein per se. She has a requirement for essential amino acids and then a requirement for enough nitrogen to make sure that all the non-essential amino acids that are needed can be synthesized. So we have to focus really if we're going to improve the efficiency and hopefully improve the economics, because if we can feed less protein in the diet at the cost that it averages, then that's an opportunity to have lower diet cost with the same production. And that obviously will result in greater economic efficiency. So in order to assess the requirements, we can assess what each tissue is doing, and that's typically how the requirement systems are developed, using sort of a factorial method. But of course, eventually we have to then relate that to supply. And so that's really what I'm going to focus on today, is how do we assess what that supply is, not only from an individual feed, but from an ingredient as well. So one of the things that we can do is we can obviously just look at the relationship between feed amino acids being consumed and the amount of amino acids coming out in milk. And through all of these, we do have to keep in mind that often we're trying to relate this to the bioavailability or the availability of an amino acid from a particular feed. And so obviously we typically can't feed a pure feed to a cow. We can for the forages, for most of them, or maybe all of them, but certainly we can't for most of the grain sources, as at least not to get answers that would be relevant to a dairy cow. So we're typically looking at trying to pull out these bioavailability values from changes that we've made in the diet. So you might have a diet, that either contains or doesn't contain soybeans, for example, or cottonseed. And then you're looking at what the differential response is between the base diet and the diet with those added to them. So if we go back then to my few arrows here, I mean, we can feed, one or more of these feeds and put them in and out of the diet, and then try to assess what kind of a response we get in milk, protein output. Or if we want to do it for an amino acid, then we're looking at, for example, methionine intake versus methionine out. Well, of course, there's a lot of steps in between the feed and the milk output, and so there's lots of things that could sort of contribute variation to this measurement. And a big one really is, is certainly a lot of the post-absorptive metabolism, has variation in it, and trying to get the cows set up to sort of be constant in terms of their efficiency of use post-absorptively as we manipulate the diet is a difficult task. So we can get around part of that by then trying to look at disappearance from the gut, because in that case we can look at digestibility or disappearance, and we can assess what the supply is based on that difference between intake and output. And that gets rid of a lot of the problems that we might encounter with variation in the post-absorptive system. But of course, the easy one to do is feces, because it's quite easy to collect. But of course, it doesn't have a lot of relevance because we have a lot of microbial action within the gut. And if you look at, in terms of what the swine and poultry industries have done, I mean, they recognize that as well. But of course, with poultry and swine, they only have the hindgut to worry about. And so they've based their systems by assessing flows that are arriving at the terminal ileum. So you can get feed minus terminal ileum as an assessment of what was digested. But we have one more problem in ruminants in that we also have a lot of microbial action in the rumen. And so to really use this method is not going to work very well for ruminants. We have to pair that really with a ruminal outflow measurement, because we have a lot of microbial activity in the rumen that's altering the composition of the amino acids. And so what goes into the rumen in terms of feed amino acid composition is not the same composition that comes out. So that requires a somewhat more technically difficult set of measurements, where we have to either omasally sample the animal to get an outflow measurement, or introduce an abomasal or a duodenal cannula to sample from, and pair that then with the ileal cannulas. And there's not been a lot of those studies that have been done in ruminants. The ileal cannula seems to be more difficult to maintain. We have actually, in our group, done, in collaboration with Dr. Knowlton, done several sets of animals and prepared them. But typically, we've found that we're not very successful with it unless we prepare them as, young heifers or young steers, and then let them, use them over time. And if you want a lactating animal, then basically you have to prepare them early and maintain those cannulas through time. So, but that certainly is an option. And another option then is to perhaps catheterize the portal vein and the arterial supply, and then you can use some flow markers to assess what the blood flow is, and measure, rather than disappearance from the gut, appearance in the portal vein. And that has similar sort of technical difficulties as the duodenal and ileal sampling. And there's not a lot of groups that have done that work, but they've certainly generated a lot of studies and a lot of information over the last 30 or 40 years using those techniques. However, going back to the sort of identity preservation, both the duodenal minus ileal disappearance and the portal appearance measurements have a reasonable amount of variation in those measurements, and the errors in terms of assessing those supplies and appearance are somewhat, relatively large, I guess, relative to the supply. And so when you then try to assess the contribution from an individual feed, that becomes a bit more challenging and problematic. Another option then is to move on down the line a little bit, so something that's a lot less technically difficult, which is simply sampling blood. And so this is easily done from a tail vein sample, or, if it's a lactating cow, a mammary vein sample, or of course a jugular sample. And one can then try to set up the animal to assess changes that would occur in the general circulation in response to individual ingredients being manipulated in the diet. And that's certainly been used quite a bit, and it has some additional challenges that we'll come back to in a minute. But if we sort of walk through those, so if I back up again and just make sure we talk about each of these. I mean, we have multiple methods of assessing these bioavailabilities, and all of them have some sort of positives and negatives relative to their use. And so what I want to do is just briefly walk through the individual processes that have been used and talk more about the accuracy and precision of those, because it really is a problem in terms of trying to identify what the bioavailability is of a given amino acid from a given ingredient in the diet when you have to feed mixed diets. So let's start with this example of simply looking at, in this case, infusing methionine into the abomasum, okay, probably via a rumen infusion line that was passed into the abomasum. And they used, in this case, a dose response model. And so they're looking at milk protein responses in relative to the infusion of differing amounts of methionine, where they had 0, 10, 20, 30, 40 grams of methionine infused per day into the abomasum. Now we know that crystalline methionine is 100% absorbable, and so there's not a digestibility issue here. We're simply looking at the response in milk to a given supply that's arriving in the gut. So it's not directly applicable to our problem, but it does highlight one of the challenges with this approach. And so in this case, they've used a 5-point response model, and they have derived a nice curvilinear response, which is sort of the expected shape of the curve. So at some point, the animal will reach its maximum potential to use that methionine in terms of synthesizing milk protein, and so it will plateau. And so if you're going to use this kind of an approach, it's really critical in my mind that you actually do a multi-point dose response curve, because it's really hard to tell, given our knowledge of the requirements today, whether or not that animal is actually set up to be responsive, and whether her responses are going to be primarily linear, or if you're going to be at the top end of this curve and you're going to have nonlinear responses. So if you use a 2-point response, i.e. 0 and then 10 or 20 or 30 or 40 grams of methionine, you have no idea whether or not you have a linear response model that you're interpreting your data with. Of course, the 2-point curve is always linear, but that doesn't mean the underlying response is linear. And the other problem with using a 2-point response is you can look at any 2 pairs of points here and see that you can have quite different results, right? So if you look at the 0 versus 10, you get a negative bioavailability. If you look at 0 versus 20, you'd get 20 or 30, you'd get very similar results, and it's a much higher bioavailability, and of course it's positive. If you use 0 versus 40, you're going to end up with quite a bit different answer. So there's a lot more variation associated with trying to assess these bioavailabilities by simply using 2 points. In the case of a dose response curve like this one, you have a curve that you're drawing inference from across the points. And so you have a lot more confidence that the curve actually represents the true underlying response, because it basically pulls out a lot of that variation that's associated with making the measurement. So if you're trying to assess a rumen-protected amino acid, for example, or an ingredient using this kind of a response model, all those points still apply. You need to really probably look at whether or not you have a linear or a curvilinear response. You should probably consider using a multi-point response model so that you can actually have better precision in deriving the information from that curve. I keep trying to go forward with my arrows on my keyboard instead of the control on the screen. But anyway, so the challenges really are the precision on this is not terribly high. And often, and this has been our case, a couple of these trials we've tried, we got no response because the animals just weren't set up to be responsive. And so getting the animals set up initially is always a challenge. And then, making sure that you can effectively and accurately interpret the data is also a challenge. And where you introduce the ingredient of interest, can slightly change the outcome as well. Obviously, we're interested in total bioavailability. It's nice to know how much is lost in the rumen and how much is not digested in the rumen. In the small intestine. But in the end, it's really just an efficiency measurement, and the cow only cares about what is being absorbed. They don't typically really care about how much is lost in the rumen and how much is indigestible in the small intestine. So feeding it is, the best approach, because then you get a complete bioavailability, and you don't have to rely on other assessments of ruminal losses to be able to interpret the data and use it. And so Dr. Tichtmeyer was on the call earlier. I'm not sure if he hung around. He's heard me talk a number of times, so I suspect he might have found something better to do. But, this is one of the few studies I think that was really done well and is using a model, an experimental model that helps alleviate a lot of the imprecision that can come with these duodenal flow measurements. So in this case, he used, I think it was 12 steers to assess these 4 different feed ingredients. So it was blood meal, corn gluten meal, fish meal, and soybean meal. And he did each of the ingredients. He fed them at basically 0 and then three levels above 0, so that he had a linear response model that he was looking at responses for. And so he measured then the flows to the small intestine. So I'm just showing this one as an example of the amount of variation that's there. If you're only looking at, again, a 2-point curve. So for example, on the soybean meal curve, each of the S's, at the 110 gram nitrogen intake level is one animal's observations. So you can see that there's quite a bit of spread in those. And so if you're looking at 0 versus 100 or 110, depending on which animals you choose, you can have quite a bit of variation. But of course, when you're doing a linear response model where you're fitting through 4 different points here with 4 animals' observations at each point, he actually got some fairly good precision on making these flow measurements. And basically, they're in between 6 and 10% variation on crude protein flow associated with each of these different ingredients. So that's quite a precise measurement and a very nice way to do this. But of course, this is just flow, so it's really only giving you information on ruminal loss. And of course, this study would cost a tremendous amount of money to do today. I'm sure it cost quite a bit then, but it would certainly cost a lot of money now to do a replicated Latin square with 12 or 16 animals, however many he used. But even with that, very good experimental design, once you then start subtracting duodenal flow of each amino acid and subtracting from that the ileal flow, your errors propagate, right? So you have a certain amount of error, even if it's only 5 or 10% at the duodenum, you have another 5 or 10%, well, probably 10% at least at the ileum. And so when you actually do those subtractions, those errors propagate. And so your errors, end up being larger than what it looked like on the last slide. And so here's his individual amino acid disappearance values. And you can see that, of course, there is differences among the 4 ingredients. And there's also differences within an ingredient based on the amino acid. But note that, all the error bars are in the 20 to, 5 to 50% range. In fact, methionine is, 50% or greater. And essentially none of the methionines, were significantly different from 0, with perhaps the exception of soybean meal, which has an estimate of, a whopping 190% availability. So it's a very good source of methionine, apparently. So when you're trying to then, this is just total digestible flow. And so when you're trying to back these things out and apply them to individual ingredients, you end up with a lot of variation unless you do a very, large amount of animals with a very strong experimental design such as this. So it's not that you can't use this method. It certainly will work. And, these are not, huge errors, but they're not very precise in terms of trying to define the difference between, for example, one soybean meal and another soybean meal. Another approach that's been used that's a lot cheaper and quicker and has had some value, I think, is an abomasal pulse dose of the ingredient after it's been exposed to ruminal fermentation in situ for a given period of time. And so the user has to decide, okay, well, would this ingredient typically be exposed to ruminal action for 8 or 10 or 12 hours? I mean, you have to pick a time. One, I suppose, could, do a time series and then dose each of the time points into the abomasum, but it's quite a bit of work to do that. But at any rate, what we typically do is incubate for 8 or 10 hours, unload wash the bags, unload the bags, and consolidate to get a large enough sample to then suspend in water, basically, and dose as rapidly as we can into the abomasum via an intraruminal infusion line. So, and it basically generates then a response in concentrations, in this case, of methionine in blood or in plasma. And so you can then do that with unprotected methionine, which is the darker line with the the black symbols, or you can do it with in this case, a prototype for rumen protected methionine. And if you then use one of several approaches that are potentially possible to integrate the area under each of those curves, you can take those two areas and divide them to come up with a bioavailability. Now, in this case, the unprotected methionine, because we're trying to keep it somewhat closer to the dose level, the effective dose level, so that we don't have large differences. But normally, if you would put in the same amount of methionine in unprotected form and also in rumen protected form, the unprotected form would have a much larger curve, higher curve, and therefore more area out of the curve, and would potentially then, might cause the animal to handle that material differently. But you can see in this case, I mean, we've raised blood concentrations by 20 to 30 fold, so it's a fairly large deviation. And so you can compare those two areas and correct then for any differences in what might have been infused in terms of the amounts of methionine to calculate the area of the curve. Now, this worked quite well for some of the earlier products that were put out that had a pH sensitive trigger, and because they released really rapidly, and so those curves that spiked up very rapidly and came back to baseline fairly rapidly as well. On these, what we found, and I was told this before, and of course I was too much time in Missouri, I guess, so I had to be shown this by myself or by our students. But essentially, the curves, as they flatten out, probably what's happening is that we're losing more and more of the baseline, so we're having more and more difficult detecting where the curve deviates from the baseline, and so the bottom of those bars that I filled in there might actually be too high, and so you end up underestimating. And the slower the release is, the more it seems like you underestimate. And so while we feel like this is maybe still useful for screening, if you're using a given sort of protection mechanism so that the rate of release is perhaps somewhat similar, you can probably use them to rank your prototypes so that you can decide that, well, this prototype's better, but the values that you get for it in terms of bioavailability are not typically representative. And so you have to actually then assess true bioavailability using some other method. And, I think, again, the earlier prototypes that released much faster, those bioavailability values that they were able to assess with this approach did match up fairly well to other methods. So I think this is really a function of having a slow release and therefore undermining sort of the calculations that we do. Probably the better one then, in terms of trying to do this with a blood concentration response, is to, rather than pulse dose, would be to either infuse in terms of developing a standard curve, or feed the product for a 7-day period to establish basically a relationship between the amount that was put into the animal and a concentration in blood that's sort of a steady state concentration. So in this case, you would, for example, infuse for 7 days at each of these different levels of methionine on the left, and that would generate then these curves where you would see an increase in the amino acid concentrations in blood, and you can see that they're linear. So as you add more methionine concentration infused, and I apologize, I forgot that these axes are reversed. So on the y-axis is what was infused, okay, which would typically be your x-axis. And on the x-axis then is the amount of the concentration in blood. So as you go up in the infused amount on the y-axis, you can see that the response in blood concentrations are linear. Now this only occurs typically if you've put the animal on a fairly high protein diet so that you're sure that they're above all of their requirements for methionine. And you can see then on the right-hand panel the same response for lysine. However, there's a lot more variation among cows in terms of their base level of lysine in plasma, but the slopes are very similar, okay? And actually, when we did a study similar to this with lysine, we saw exactly the same thing. There was a lot of variation among cows, but within a cow, it was a linear relationship. So, cow effects then are important to make sure that they're taken out. And so if you're going to, to do this kind of an approach, you have to make sure that you're comparing within cow, particularly for lysine. And so when you then reduce those slopes, using the standard curve to actual bioavailability values, and this is from that same publication, they basically did end up in this particular publication with some fairly tight standard errors on the methionine bioavailabilities, whether it was by itself or as a co-product with lysine. And so I've circled those in red just to point those out. And so you have a good precision with that method, and basically it matches up to all the other methods in terms of this product, and so it provides a reliable and accurate and precise answer. However, note on the lysine, it was done the same way, but the error was quite a bit larger. And in our study, which we didn't publish, we saw the same. We didn't check methionine. We didn't use the approach for methionine as well. We just looked at 3 different prototype encapsulates for lysine, and they did, vary. At least one of them varied from the other 2. But the standard errors were all in the 16 to 20% range. So essentially the same as what Rocan found on his study. And we did another study, using the same approach and tried to refine it, and we ended up with the same 18 to 20% error on that. So I think, using this method, you can anticipate, perhaps better, reasonable precision with methionine, but seemingly not so for lysine. And I don't really understand why there might be a difference in those 2. Of the 2, methionine typically offers more challenges in terms of the amino acid analysis accuracy or precision than lysine does. But and of course I want to emphasize that these dose response or the standard curves for interpreting the unknowns were multi-point standard curves. And so the precision in generating that curve is quite high. There's a very small standard error of the prediction for those curves. But in application, then a lot of people, and this is done, in the poultry industry all the time, is they'll then move to a single point comparison. So you're going to pick a 1 point off of the curve. Once you know the curve is linear, you're going to do another study, and you're going to include in that study 1 treatment for 1 point on the curve versus your standard and 1 point of infusion. And then another treatment will be an ingredient of interest, right? And so you end up with single point comparisons. Well, the curve now has, or your standard now has the same kind of error as your product does. And so you end up with we'll end up with a much larger standard error of measurement and prediction from that approach. So I would guess if you're using single point comparisons where you don't have a multi-point standard curve, your precision is probably going to be 25% or less, okay? I mean, it's going to be a much larger standard error of measurement. So in essence, it's not any different than what Dr. Titkemeyer observed in his PhD work. So the last approach then, or well, I guess the next approach I want to talk about is trying to use some kind of a marker rather than measuring the flux itself, okay? Because the flux measurements are difficult to make. And so I want to introduce this just by a little schematic here to sort of give you the idea of how a dilution works, okay? And so let's say that you wanted to know how many cars were going down this, rather large freeway in LA. Well, right now there's none hardly going down there, but on a normal time, there would be a lot of cars on the freeway in LA. And you can't really easily count them. You can't observe them directly. And so what you can do, though, is you can sample those cars. You can take a sampling of 10 cars and find out, what color each of the cars are. So if you have, if you observe that there's no white cars, or you observe how many white cars are normally there, and then you line up a bunch of rented white cars on the on-ramp, and you start having one of them drive onto the freeway each minute, after a little while, you can then sample downstream and figure out what the ratio is of white cars to colored cars, right? And you can correct then if there was any white cars that were in the stream before you introduced. So in this example, if I count my cars downstream in the bracket, I've got 10 colored cars and 5 white cars. I know I introduce 1 white car per minute, with my rented car stream. And so I can divide that out and find out that, well, there must have been twice as many colored cars entering that or on that freeway, i.e. I must have had 10 colored cars to get the right ratio, right? And so I must be having 2 cars per minute that are going down that freeway. And so you can do the same thing then, if you change the number of cars going down the freeway, but you hold your white car entry the same, you basically can derive by difference. Now, what I want to point out is that my white to colored ratio is 0.5 when we had less cars on the freeway, and it's 0.33 when we have more cars. So these are reversed, right? I mean, when we have a larger dilution, that means we have more unknown material entering, okay? And so this is the same principle that Bill Weiss and Norman St. Pierre used for assessing methionine bioavailability using selenium as a marker, okay? And so what the selenium is not just selenium, but it was selenium that yeast had incorporated in place of sulfur into the methionine, okay? And so they're capable of doing that, and they don't seem to discriminate much against that. So if you grow up a batch of yeast with with selenium in the media, they will incorporate a fraction of the selenium, or a fraction of their methionine will have selenium in it. So if you feed yeast all the time, which is the equivalent of my white cars entering the freeway, you can then look at plus and minus your product and see how much dilution you get. So when it gets diluted, then that means there's more being delivered by that product. And so you can work out what the bioavailability is. And you can see from their error bars on this that it's, reasonably precise. It has about a 10% error. So we're probably twice as good as what we can achieve by a flux measurement. And there's really no clearance. I mean, there's no problems with this method, right? It's not subject to having a linear response in milk protein. It doesn't have the measurement errors. It doesn't require surgical intervention. So it's a fairly simple way to do this. The problem is that we only have methionine that we can do it with in terms of selenium. So we need other markers. Well, fortunately, we do have and can purchase carbon-13 labeled amino acids, and we can measure those fairly precisely as well. And so we end up with being able to infuse those and use the same approach. And this really derives from some work about a dozen years ago from Hélène Lapierre's group. And they sort of did it the opposite of what Norman and what we would use. And they tried to look at a dose bolus at the beginning and then look at the decay curve over time on the plot on the right. But the problem with that is it is a challenge because they gave a huge amount of the isotope, so it costs quite a bit of money to dose that, and it's all gone basically in 30 minutes. So you have a very short window to be able to assess that over. And so what we've done is turned it around and said, okay, well, let's just start infusing it at a constant rate, and we'll look at the rise to plateau. Now, ideally, it would be real simple math if we just infused long enough to get the animals to plateau. but that would take at least 36 hours, and that's a lot of isotopes. So we essentially basically do a shorter infusion. We're doing 8-hour infusions now, and then we use a bunch of math to sort of predict what the plateau would be. So we're substituting math, for isotope costs. And so in this plot, which was published by Shinbei earlier this year, I think it was, we have a range of sort of precision here. So it's different ingredients that were assessed using that method. And if you focus first on the distiller's grains in the middle of the plot, you can see that the error bars are roughly about 10%, and the same for the brewer's grain. Now, all the other ones have higher errors because the error is an absolute error, right? And so the rest of those ingredients contribute very little to the protein supply of the animal, and we substituted or we added 10% of each of the ingredient to the diet. So when we added 10% distillers or 10% brewers, we added quite a lot more protein than when we added 10% soy hulls or 10% corn. So those errors sort of increase in those because we probably should have provided more of those ingredients when we did these assessments. But again, just as Evan saw with his work, there's differences among the amino acids in terms of their digestibility, and there's also, of course, differences among ingredients. But this method then offers us a way to have identity preservation within a mixed diet without having to load the diet up, to a high degree with that ingredient. And provided we can, provide enough protein, we can get some reasonably good precision out of these methods. So it's a good way to do that assessment. So the last method then I want to quickly touch on is this. It's a derivation of Carrie Estes' work that was published before Shinbei's. And in this case, the two blue bars are from her assessment using that method, which she's the one that worked out using that. And they were of blood meal and feather meal using the same approach. And you can see in this case, we contributed a lot more protein to the overall diet, even though we were only able to feed 2% of the diet. And so the error bars have shrunk considerably. So we have really high precision in this measurement for these two particular high protein ingredients. She then subsampled the blood meal and the feather meal and sent off between 10 and 15 samples to each of two labs for two different assessments. Each of the labs did modified 3-step as published by Ross a couple of years ago. And then one of the labs, also in addition to doing the Ross method, just used the standard 3-step method. And so what I want to do is just point out that the first lab, in terms of blood meal, got an answer with their sort of in vitro assessment that was very similar to what we measured or what she measured in the cattle, okay? But you take the same method and the same ingredient, because these are just subsamples out of the same supply that she used in the experiment. The other lab got a considerably higher number. Now, both labs have reasonable precision on making that measurement for the blood meal. And when they used the modified 3-step as an older method, they got similar to the second lab in terms of the Ross method, and it had pretty good precision as well. But of course, it's 20–30% over. And so then we just as a comparison, we got the Cornell models library values. The challenge then is when you look at the feather meal is we got the value in blue from the animals. Neither lab were able to get a reasonable estimate for the feather meal using the Ross method. And the second lab 2, had a really high error of measurement across the 10 samples that we sent. And the older method overestimates. So again, Cornell has about the right value, but the methods, in terms of in vitro, are pretty suspect here. So in summary then, there's multiple ways to assess amino acid bioavailability. If we look at it from an accuracy and a precision standpoint, where accuracy is, are we getting the right answer on average, and precision is, what's the error of our answer. In vitro, I think are extremely biased in terms of the methods we have today. I don't think you can trust them for anything, and perhaps they can be trusted within one lab, but they don't seem to be able to be moved across labs to get reliable answers. The pulse dose method, underestimates when there's slow release. The milk protein response, should be good, but it can be nonlinear, and so it can generate biased answers if you don't actually do a full dose response curve. And the jugular infusion, with the isotope works fairly well, but we do have a slight bias we have to correct for. In terms of precision, the intestinal disappearance is fairly low. The in vitro methods are fairly high, high precision within a lab, but low precision across labs. The milk protein response has somewhat low precision. It can be sort of moderate maybe. Blood concentration increases using a linear response curve, are moderate precision. The pulse dose is moderate precision, and the tracer dilution can be high precision if you can provide enough amino acids in the diet. So with that, I'll close, and I did run a little bit over. My apologies.
All right, let's get to some questions here to wrap up the day. So you touched on this a little bit with that last slide. What's your opinion on using in vitro procedures to provide that amino acid bioavailability estimates for rumen-protected amino acid products, and what are the pros and cons in that discussion?
Well, I don't think any of those, until we actually demonstrate, with in vivo measurements that the in vitro is giving the absolute answer. Okay, so we got to divide the answer into accuracy and precision. They probably aren't accurate, and we have to assess in vivo to determine whether they are before we can trust the accuracy. But the precision is probably good within a lab. So if you wanted to rank protected amino acids within a lab, they might work, but they really haven't been looked at. So I don't really know how well they will work. I think we have to do more work on those. Obviously, we want to get to an in vitro method for all this stuff. We can't be doing these large animal trials on everything, but we have to, benchmark those in vitros against in vivo and make sure they actually do work.
So when you consider current and past rumen-protected methionine feeding studies, how much did variation in dietary sulfur and cobalt intake or microbial methionine synthesis and delivery play roles in the response? So, and expanding on that a little bit, don't metabolism and synthesis of various actors in the single carbon transfer world depend on cobalt for microbial B12 synthesis and a source of sulfur, especially for animals grazing in the pasture?
Well, most of that question is outside of my area of expertise. I think Heather or somebody would be better to answer that question, but I can speak, I think, to the first part of it, which is we know there's variation among animals, right? If there was no variation among animals, we wouldn't be able to be genetically selecting for animals. And so, our assessments that we've just finished in terms of looking at the variation that's contributed, by a combination of the environment, right? Because when we're making measurements across animals and across locations, we've got environmental variation. It's in the 12 to 15% range, right? So depending on what animals you chose, and among your animals, you can get quite variable answers. So there's no real way to control that other than using Latin squares and things like that, where you can control some of the variation within an animal.
Dr. Hannigan, we had quite a few questions come in here right at the end, but friends, unfortunately, we're out of time, and so our presenters and some professionals from Balchem's team of experts will follow up on some of these individual questions after our virtual symposium concludes. But I want to thank Dr. Mark Hannigan with Virginia Tech University, along with all of our other presenters,